physician, and I'm based at various facilities, public health facilities here in Melbourne. And I also work at the Burnett Institute, and I am a committee member at ACNET as well. So I'll tell you very briefly about Burnett. So Burnett is a medical research institute and an accredited international NGO. And we do research and health program development in the Indo-Pacific region, as well as in Africa. And we're based in Melbourne, Myanmar, and PNG, primarily in Melbourne. And I particularly belong to the TB working group. So we generate knowledge and solutions to NTB in Asia Pacific. And we have, April and I have recently, as well as Phil, who's can't be here today, Phil Ducrot, we've recently started an Asia Pacific clinical trial network called TB Aspire to facilitate collaboration. And this is one of the collaborative efforts. So thank you everyone for coming today. And if you'd like to learn more about TB Aspire, I'll put my email in the chat and you're more than welcome to email me anytime.
Thanks, Gail. So I'm co-chairing with Dr. Cross. So I'm Andrew Burke. I'm a infectious diseases and respiratory physician at Prince Charles in Brisbane, and I'm current chair of the steering committee for ACNET, which, as you can see, is the Australasian Clinical TB Network. And so just briefly what we are is the idea is a clear network across different specialty groups. We have representatives from TSANZ, which is Thoracic Society, as well as ACID, Australasian Society for Infectious Disease, and Australian Respiratory Council. And so really just trying to get people to know each other. The TB world's not that big in Australia, so it's nice for us all to know each other and collaborate where we can, and we're trying to promote educational activities such as this. Membership's free, and we might drop that in. I'm sure Kai, our administration officer, will drop in the link if you are interested, and then you'll be notified about these, which we hope to be a regular event. And this is the first one we've done. So so I might just kick off, Gail. Is that all right if I welcome Chris? Yeah, so look, on that note, I'd just like to welcome and thank Dr. Chris Coulter for agreeing to be the first presenter for our journal club, hopefully the first of many. So I first met Chris probably 20 years ago. He was the director of infectious disease at Prince Charles Hospital. I was his registrar and been a great mentor to me and many other people here in Queensland. He's director of, as well as being ID physician, he's microbiologist and director of the Mycobacterium Reference Laboratory in Queensland, which is also a WHO collaborating centre. And he's also a medical advisor for TB and communicable diseases in the communicable diseases branch of Queensland Health. Chris is on WHO technical advisory groups for microbiology as well. So thanks, Chris. I'll hand over to you.
Thanks, Gail, and thank you, Andrew, for that kind introduction. And if I have managed to do any mentoring, that's a real plus. So look, without further ado, I'll share screen. And I'm going to assume that you can see that. Yep. Okay, so Gail suggested I review a paper that was a nice, juicy lab one, and then I thought, that's pretty interesting, but I'd like to do something that with a bit of therapeutics in it as well as lab. And I think this paper probably hits the mark. So this is a paper that's in press currently in the American Journal of Respiratory and Critical Care Medicine. It summarizes work that has been done over preceding years, as I'll demonstrate. And the title is High-Dose Isoniazid Lacks Early Bactericidal Activity Against Isoniazid-Resistant Tuberculosis Mediated by katG Mutations, a randomized phase 2 clinical trial. And Kaman Kuala Gausi is the first author, and then there's obviously a big team of investigators for this, which was for this clinical trial. So just going to the abstract. So the rationale for this study was that what they call observational studies suggested high-dose isoniazid may be efficacious in treating MDR-TB. So this probably perhaps can be demonstrated initially by thinking of the so-called Bangladesh trials. So the Bangladesh regimen was a sort of 9 to 12 month 7-drug regimen, which was efficacious against MDR-TB, and it was a trial where they sort of whittled away at different regimens to come up with what they thought was the most efficacious regimen, and it included high-dose isoniazid. And those findings were replicated in demonstration studies in Africa. There's also been studies from India supporting the use of high-dose isoniazid in an MDR regimen. So these authors, as I'll tell you about, have already demonstrated that, in fact, for low-level resistance mutations, isoniazid in increased dose did have activity against TB in patients, but they were particularly wanting to look at katG, because a lot of those demonstration trials actually didn't tell you whether they were looking at low-level or high-level resistance, and katG is the high-level resistant marker. The Bangladesh one, as I understand it from personal communication from Armand van Doorn, probably was mostly katG mutated, but Armand is no longer with us, and just to quickly point out for those that knew him, what amazing things he did for TB on a global platform. But I'm digressing a little bit. So back to what was this study? So it was a registered clinical trial, a phase 2A study, so looking at drug dosage and safety. It was randomized and open label, and it got patients with drug-resistant TB with isoniazid resistance due to a katG mutation, and gave them not 5 milligrams per kilogram, which is the usual isoniazid dose, but 15 or 20 milligrams per kilogram for 7 days. And they looked at quantitative measures of bacterial burden each day of those 7 days, and then did a lot of intensive PK sampling on day 6. And using some modeling, set about to try and find out how much of an early bactericidal effect isoniazid gave when used in high dose in this category of patient. So just a few quick dot points on isoniazid. It rapidly reduces TB bacterial load in the first few days, therefore lowers the risk of transmission, and for those in hospital-based practice, requirement for prolonged isolation. It protects the companion drugs from resistance development, and worldwide, it's about 11%, sometimes a bit more, of new TB cases, which might be either isoniazid mono, poly resistant, or multi-drug resistant, where co-resistance with rifampicin is essential for that categorization. Now, it's a prodrug, so it does require activation in the body, and that's done by this catalase peroxidase enzyme, which is encoded for by the katG. And that activated drug then binds to NIL-ACP reductase, and that binding interferes with mycobacterial cell wall synthesis. Now, upstream of the inhA gene itself is a promoter area, and that includes the so-called FABG1 gene. And this promoter area, if it's mutated, it dysregulates the production of NIL-ACP reductase. In fact, it usually increases the amount that's produced, which then overcomes the effect of the drug, because there's a lot of target that it's got to, that it sort of swamps it. Generally, katG mediated resistance accounts for the majority of isoniazid resistant TB, including MDR-TB. So, as I said, they had already tested the hypothesis that they could overcome sort of resistance for low-level MICs, and I just wanted to point out from the text of the article that, in general, isoniazid susceptible strains have a minimal inhibitory concentration of 0.03 to 0.06. If there's an inhA promoter mutation, then it's typically low-level resistance, say of 0.2 to 1, and katG mutations give high-level resistance, which might be 2 to 8 if there is, I suppose, presumed partial loss of katG, and if there's complete loss of katG function, then greater than 16. As far as the PK/PD metric of bactericidal effect, it's most closely correlates with AUC to MIC ratio. And another important factor in considering the effects of isoniazid is how it is metabolized. And it's been known for quite a long time that we have sort of fast metabolism and slow metabolism. And in fact, this is tied to the N-acetyltransferase 2 enzyme. And the sort of the balance of what we now categorize as slow, intermediate, or rapid acetylation is it varies geographically. So if someone is metabolizing slowly, then generally the exposure and area under the curve will be greater. And this is from not this paper, but from a different paper, which was a review of NAT2 activity in different populations, just to show you how it may differ, that in general in Africa, slow acetylators predominate as they do in Europe. But in Asia, it's only a third of the population that is generally said to be slow acetylators, with a higher proportion that are rapid acetylators. Now that's sort of on a continental basis. Then you can drill down into different ethnic groups and different countries and get different answers. But the important thing is that there is a genetic basis for how quickly you metabolize isoniazid. So back to the study. It was part of the AIDS clinical trials group's studies. It was called Iron Hindsight. So the INH there being short for isoniazid, and I've already said that it was a phase 2 open label study, but it was looking at only pulmonary TB, so not extrapulmonary TB. And in this study, it was from 2 centers, 2 countries, Haiti and South Africa. And it was, of course, approved ethically with written informed consent. Now, I started reading the paper and then got up to sort of methods and details of the study design were previously reported previously, and the results of the isoniazid sensitive and inhA group previously published. So there's this reference 20 and 21 cited a lot, because so therefore you have to really go back to the earlier papers to work out what their methods were. And these are the 2 papers that appear as reference 20, which was published in 2020, and reference 21. The reference, this one here, the semi-mechanistic model of the bactericidal activity, etc., is mostly a methods paper to look at how they came up with some of their modeling, and the sort of the more substantial results paper was this one here, where they looked at not katG, but inhA mutation and compared it to non-mutated strains. So that earlier study, so reference 20, is summarized in this flow here, where they recruited patients between July 2014 and 2017, whereas the katG group, and that was just in South Africa, not Haiti, whereas this katG group was from 2 countries and recruited between 2019 and 2021. So the onset of both these studies predates perhaps what we now would reach for in our sort of regimen selection for MDR-TB. But at the time that these studies were done, the big change had been the 9 to 12 month short course regimen that used high dose isoniazid. Now, what did they do? So they cultured, the patients actually had to be quite productive. They had to produce more than 10 mils of sputum a day. And they did daily sputum cultures. In the first study on the inhA, they looked at a bacterial kill model where they actually did colony counts on solid 7H10 agar. They've still got it in the supplementary material relevant to the paper we're talking about today, but in fact, they didn't actually do this part. What they did do is they set up use the same pharmacokinetic model, which accounted for both liver metabolism, the NAT2 genotype, and they determined kill efficacy. And they looked at time to positivity, TTP, in the MGIT, so Mycobacterial Growth Indicator Tube model. So it's basically this logarithmic growth, how long until the machine went ping and indicated that you had positive growth in the test tube. Now, a couple of points here to make is that they looked at drug susceptibility in the first study by using 1% agar proportion. They should have, but they didn't actually specify which agar. The two common ones, 7H10 and 7H11, but it wasn't actually stated. And then for the katG study, they in fact use a totally different method, which is a broth microdilution using the commercially available Sensititer plate. It's not yet WHO endorsed, and there is no agreed cutoffs for determining susceptible and resistant. That is in process. There's a lot of work happening on that at the moment, but at the time of the study, it was not as well established as the agar proportion method. Now, how do they determine katG and inhA? Well, they relied on the Hain line probe test, and this looks at a small number of specific mutations for RPOB, and in the context of this paper, katG and inhA for isoniazid. And in fact, in the paper, they say that it targeted inhA. It actually doesn't. It targets the promoter region, not the coding region. So all these mutations are upstream of the actual inhA gene. And there's really only 3 mutations they're looking at for inhA at positions -15, -16, and -8. And there's really only 2 positions for katG. And most of the world's katG is at position 315. But nonetheless, as per the heading of this paper, published in 2017, that when you look, you'll find resistance conferring mutations that fall outside the targets of these commercial assays. And of course, now we rely on whole genome sequencing for a comprehensive analysis of the resistome. So the point being is the reliance on the Hain test may exclude some mutations which are conferring resistance because they're just not picked up, and therefore, as an example, specimens we get from Papua New Guinea usually only have an inhA promoter mutation, and that equates with low-level resistance, but they're usually high-level resistant, and it's because they have a coding region mutation that coexists, and that's not in the commercial assay. You only get that if you do targeted sequencing or whole genome sequencing. From the pharmacokinetic pharmacodynamic model, now they used non-mem, and I'm going to tell you right up, I don't know what that software actually does, and maybe others do know what it does, but that was nonetheless the software they used for their modeling. They looked at isoniazid PK with a 2-compartment model, and as I mentioned, they took into account NAT2 genotype and its effect on clearance. And they looked at the early bactericidal effect, assuming a sort of sigmoidal efficacy max function. And they looked at the effect of the katG mutation on potency. So this is EC50. This is the concentration required to exert 50% of maximal kill. They looked at delay in onset of growth, the growth rate of TB in the MGIT, and the baseline bacterial load. Or at least they did in the first study. In the katG study, they didn't look at baseline bacterial load because they didn't do those colony-forming unit counts. Now, this is the formula that they used to determine the bactericidal effect, and I'm going to also admit that I wouldn't be able to tell whether this is the correct formula or not. But I do note that when they're looking at time to positivity, so they didn't use colony counts in this study, only time to positivity in the commercial MGIT, but they did try and account for the fact that there might be different growths in the first couple of days. So they looked at a parameter that covered day 2, and also then a parameter that covered from day 2 to 7, as well as the total time to positivity over each of those days. So the actual positive event occurs many, many days later, but they know which day that they collected the sputum on. As far as our results go, So first of all, the demographics, they're all similar age. So note that they're referring to the earlier studies here when they're looking at the isoniazid susceptible and the inhA group. The katG group, I've already told you they used 2 different high concentrations of isoniazid, but in the inhA study, they used 5 mg per kilogram, 10 and 15. About 20% overall were HIV positive, and that was consistent over all those different study arms. They had to be smear positive to get into the study, and they couldn't be scanty. They had to be 1, 2, or 3 plus smear positive. And you can see a little bit of difference in that the smear grade, the inhA group were more likely to be 3 plus smear positive or heavily smear positive than the katG group. There's no stats there as whether that's significant. The other thing is that looking at all these results, you've got 2 arms in the katG group, but there's actually no comparators, perhaps because the numbers were only 10 and 11 in each group respectively. But we're assuming that there's even matching between the 10 and 20, because I don't think that was actually spelt out in the paper. Body mass index relatively low. Cavitary disease was common, but it was more common in the inhA group than the katG group. And the bacillary load, as I said, they couldn't tell for the katG group in this study, but in general, the baseline bacillary load was higher in the inhA mutated group compared to the wild type group, so about a log heavier TB burden in those specimens from the low-level resistant patients. And then they looked at time to positivity. So while the sputum TB burden was higher in the inhA group, the time to positivity was longer. So in general, the greater number of viable, healthy bacteria you've got, the shorter the time to positivity in that liquid culture system. And then, as you can see in this, looking at particularly at the katG group, about 20% were rapid acetylators, but most of them were either slow or intermediate grade. So what did they find? Well, first of all, they neatly demonstrated that area under the curve correlated with the dose given, although the confidence intervals look a little tighter with the 5 milligram per kilogram dosing, with a bit more variation at these higher doses. And here's the main result slide of the study. So what this is looking, it's basically using that formula that I admitted I couldn't fully understand, to take time to positivity to a measure of early bactericidal activity. I should have mentioned, of course, that all these patients are getting isoniazid monotherapy, something that you wouldn't do clinically, but it's felt that over this 7-day period, it would not make them more likely to become resistant or compromise their therapy. So it's a sort of a short single drug study. And after that 7 days, they go back onto standard programmatic treatment of TB. So this is the early, my alarm goes off. Is that 20 minutes?
I think it is, Chris, but keep going. It's fine.
Keep going. Okay. All right. So this is the early bactericidal effect for drug susceptible TB at a dose of 5 milligrams per kilogram. 5 milligrams per kilogram. And you can see that when you use a dose of 10 and particularly 15 milligrams per kilogram of isoniazid, then your early bacterial effect for inhA mutated strains is about the same as it is for fully drug susceptible wild type strains. But if you look at the katG, there is in fact very little early bactericidal effect, and this is sort of logarithmic, so it's sort of at least 10 times weaker than what is seen in inhA. And so even at this high dose, it's not zero early bacterial effect, but there's not much bactericidal effect. So I've just said that. The other thing is that they observed a weak positive relationship between MIC and the EC50 And really, they could only get a good correlation of resistance when they, of efficacy when you combine the genotyping, the NAT2 data. In fact, they felt that NAT2 data was more useful than the MIC. This is some of their modeling, which shows that in the light blue, this is 5 mg per kilogram. This is the EC50 for it's actually EC80 for a drug susceptible strain, and 5 mg per kilograms clearly above that, but it's not really cutting it for a inhA mutated strain, and definitely not for a katG. And with their modeling, they predict that even at 20 milligrams per kilogram, you wouldn't get to the EC80. This is saying a similar thing with looking at sigmoidal fraction of the maximal kill, and clearly you need a higher effective compartment dose of isoniazid to try and get close to the EC80 for a katG mutated one, which I've just said you're not going to do. This is some modeling and some observational work looking at the sort of the time to positivity, and for the inhA colony forming units reduction with different dosing. You can see that with drug susceptible TB, it didn't matter what the NAT2 status was. They all seemed to behave the same. But when you start to see divergences of these performance curves when you have drug mutation. You can also see that the starting time to positivity in the drug susceptible is substantially lower than the drug resistant ones. So the actual curve starts at a different point. I'll move on. So I've already said that they observed this weak relationship with MIC, and I'm going to come back to that in a sec, and we'll quickly go to discussion. So their main conclusion of this 7-day trial was that there was little or no early bactericidal activity observed for participants with MDR-TB where they had katG mediated resistance. And this was in contrast to their findings with the inhA mutated bacteria. I didn't really show it on that slide with multiple graphs. There was some activity of the, perhaps I can show it. So at the blue, sorry, because we're running short of time, I didn't explain this very well, but blue color is slowest acetylators, green is intermediate, red is rapid, and there was some impact to time to positivity if you had a slower acetylator at this high dose. And that's what they modeled, because they didn't do colony forming units, but they modeled the colony forming units, and that shows it a bit better, where you can see the rapid and the intermediate acetylators are hardly budging off the horizontal, but you do get a bit of an effect if you got the big dose and the slow acetylation status. So they felt that for isoniazid to have any meaningful EBA in the presence of katG mutation, you'd have to use doses much higher than 20 milligrams per kilogram. But that's a problem for another reason, because we know that in this slow acetylator group, we're more likely to see side effects even from conventional dose isoniazid with peripheral neuropathy and hepatotoxicity. They point out in the discussion that these findings are at odds with the things like the Bangladesh and subsequent short course trials showing an effect of isoniazid for the beneficial treatment of MDR-TB. And then they go on to discuss some of the reasons that might be so, but really without evidence of partial activity of katG and so forth. They said one unanticipated finding of this study was the MIC values overlapped a lot. So they found not those MICs that I mentioned earlier, but the sort of median MIC for susceptible TB at 0.2, but with a range up to 1 for inhA and 4 as a median, but with a range down to 0.03 for for katG. Bear in mind the katG was done by the broth microdilution. So they felt without including the NAT status, then the effect of MIC on kill was not observed, which is a little bit odd. And one of the reasons being is I'm not sure that they actually did the MIC testing properly. If you look at this WHO and find technical report, they looked at a whole lot of studies on trying to find an ECOF, which accounts to pretty much the critical concentration. So 99% of strains are likely to be inhibited in 7H10 media by a concentration of 0.2. So you don't expect 1% to be above that and being wild type. And they found 0.2 as the bottom of their range for drug susceptible TB. And a similar figure if you're using 7H11. I didn't reference this. This is from the Cryptic Consortium, where they're suggesting for the broth microdilution that susceptible is 0.1 and then 0.2, 0.4 is a borderline range, which really accounts for most of the inhA mutations. They also made some assumption about fitness. So inhA mutated bacteria grow more slowly. Sorry, I'll start that again. inhA mutated bacilli grow slower than isoniazid-sensitive bacilli due to microbial fitness. And the model assumed that inhA and katG mutated bacteria would have similar rates. And I'm just not sure if that is true, because fitness does vary with different mutations, not just for the gene you're interested in, but all the other genes that help define an MDR phenotype. And it also, you need to take into account compensatory mutations. And so I don't know if it affects their final conclusion, but I think that assuming the fitness between a mono-resistant strain and an MDR-resistant strain is the same is not necessarily true. And as I said, despite those sort of minor, I suppose, criticisms of the method from the laboratory point of view, the final statement, I think, probably still holds that even high doses of isoniazid aren't going to really cut it for katG mutated strains of TB. And that little group of slower acetylation, high dose, probably variable MIC where it's on the lower side, perhaps when the stars align, then you do get an effect. So brighter minds than I have considered this paper, and if you go to the journal, you can read the editorial. And so where does it leave us in our sort of thinking? So first of all, that suggests some interesting findings, suggests that maybe isoniazid doesn't have a role for katG mutated strains. But do we still need INH for MDR-TB now that we have the bedaquiline, protonamide, linezolid, moxifloxacin regimen, so-called BPARM? If we take it the other way, do we need levofloxacin for low-level isoniazid-resistant monoresistance? So should we really just be using a standard regimen, but with high-dose isoniazid in that 10 to 15 mg per kilogram range, or all 4 drugs for 6 months? And I didn't refer to the safety data, but they didn't have any major events that they could attribute to the drug in the 7 days. But I think everyone who treats TB would expect that you might run into problems with isoniazid a little further down the track than 7 days. And look, because they're relying on growth, whether it be in solid media or liquid media, as soon as you start processing a TB sample, you bathe it in sodium hydroxide, so you kill off a whole lot of TB anyway. If there's variability between the batches and the runs, you might kill off variable number of TB. So there's a lot of noise in that. So should these studies really be replicated with non-culture methods? So for instance, the TB mycobacterial load assay, I don't know if it's commercial yet, but it's certainly based on messenger RNA. So you've got to have viable TB, and it's quite good at quantifying how much viable TB is in a sample. And should we be looking at the whole resistome with whole genome sequencing rather than the sort of blunt instrument of the Hain line probe test? So I'm sorry if I went over, but I think there's still plenty of time for discussion. Thanks.
Thank you so much, Chris. That was fantastic. a really lovely overview of the paper and really great insight into the laboratory side of things. Shall we start off with questions? So anyone with questions, please put them into the meeting chat. We certainly want this to be collaborative in the true journal club sort of sense. The more questions you ask, the more robust the discussion and enriching the discussion can be. So Sadid asked a question. Hi, Sadid. Have never prescribed high-dose isoniazid. Is there a sense of how well it is tolerated? Is it routinely given with a higher dose of pyridoxine? Query 100 milligrams. My response was I've never prescribed more than 5 milligrams per kilogram, but I'm sure there are people here in the audience with far more experience treating TB.
I can make some opening comments on that. So usually with the high dose isoniazid, say for one of those sort of 9-month drugs where it's sort of 7 drugs going down to 4. So usually about 600 milligrams for an adult. So it's, I guess, sort of 10 mg per kilogram type ballpark. We always do give it with pyridoxine. I don't think you need to give 100 milligrams of pyridoxine. It might start to be counter efficacious because high doses of pyridoxine can, I think it's compete with isoniazid for I don't know if I'm explaining this correctly, but it basically can counter the effect of isoniazid if you've got too high a dose of pyridoxine. So I would still probably, see, because in that longer regimen, you've also got them on a thiamide, so they're probably on prothionamide as part of that regimen. So at least for MDR, as opposed to what you might sort of postulate you could use for monoresistance. So you probably use about 50 milligrams of pyridoxine because they're on both the thiamide and the high dose isoniazid. But I just think you've got to be a little bit careful with big doses of pyridoxine.
I did notice in the editorial that accompanied the paper, Jerry Davies made reference to this small trial, which compared 5 milligrams of isoniazid versus per kilogram versus 16 to 18. And in that paper, they did notice a higher risk of peripheral neuropathy at the higher dose compared to the regular dose. And I've put a reference to that paper in the meeting chat. But I think also, and maybe Andrew, you could quickly speak to this. I think there's a risk of having a increased risk of DILI with a slow acetylated status. Is that true? That's what I remember.
Yeah, that's right. So I see Kai's got his hand up.
Yeah.
Oh, their hand up. Sorry. But yeah, so basically, just briefly, so the acetylated status. So there's no gene test in Australia for that. It has to be sent away. So one of your questions, Gail, we had before this meeting was about whether we should be doing, so-called personalized medicine and trying to determine up front in high-risk patients maybe, or people considering high-dose therapy on about determining their acetylation status. Nowhere in Australia. I discussed with our genomics lab in Queensland, and they said they were open to developing it, but then when I did a bit of a straw poll of TB experts around Australia, none of them felt they'd really act on it. Okay. So what we do have in Queensland Health is we have an isoniazid assay, but we've also developed an acetyl isoniazid assay, so you can work out the ratio and then work out in that way what their acetylation status is. And so caffeine, you could also do that because caffeines have metabolized by the same pathway, and so caffeine is also used as a surrogate. So we're going to write a TDM document for Australia, and we'll include that in a lab this year. I see Dr. Gray's online. This a little job for the second half of the year. So the question then was about, so slow acetylators will have they metabolize isoniazid slowly, so they have higher drug concentration, so they're more likely to get side effects. And that's been fairly well shown in different studies around the world. Probably more associated with drug-induced liver injury than peripheral neuropathy, but there is a correlation with both.
Kai, your question, do you want to turn your mic on?
Oh, no, it was just more a response to that question about the high-dose isoniazid. So we don't have a lot of great data, but we used to use a bit of high-dose isoniazid in patients in PNG, some time ago, particularly with individualized regimens. And as you mentioned, Chris, we're aware because of the work out of your lab that it was predominantly a clone with that sort of inhA promoter mutation and the actual inhA mutation. We didn't have really good data on the sort of grade 1 and 2 adverse events, but just anecdotally, we had a few pretty bad adverse reactions with the high dose isoniazid, and I think that there was an issue actually with the WHO PMDT high dose isoniazid dosing table, where they were giving sort of weight bands and suggested doses that probably exceeded or above that sort of 20 milligram per kilogram range. So we had a couple of patients who had isoniazid overdose when we're treated according to those guidelines, and that sort of presents as really refractory intractable seizures and status. And the antidote we sort of figured out on the go was using high dose pyridoxine. It sort of was able to sort of neutralize that. But
thank you, Kai. Useful. Chris?
No, I was going to say, look, I just wanted some validation that my statement that there is you can have too high a dose of pyridoxine is correct.
Can I comment on that, Chris?
Yep.
So look, I was on the therapeutic guidelines writing group for TB, and Justin Waring from Perth said pyridoxine should be taken off as a routine because it can actually make worse the peripheral neuropathy side. And I actually looked back through it, and CDC and the British guidelines have also taken pyridoxine off as a routine sort of supplement. And one of the rationales when I looked into it is that you only need 5 milligrams of pyridoxine a day to meet your dietary requirements. So giving 25 to 50 is actually already a high dose. And so it's actually quite easy to get that 5 milligrams. So very few people would actually be deficient. And when it was sort of thrown in was when the early studies in India and southern India, which the British Medical Research Council ran, there was a lot of beriberi around back then. There's a lot of malnutrition, and so I think it was added in historically for that reason. And then to come down to, I think what you're alluding to, Chris, is that there's actually 3 different formulations of, you'll be sorry you asked this question now, you're probably getting more detail than you wanted, but there's 3 different versions of pyridoxine in our body. And the one we give is, it competitively antagonizes the other more natural variations. And there's actually can paradoxically worsen peripheral neuropathy if you give high doses. So it's a bit complex, but it's not just as easy as increasing the dose. And anecdotally,
does it interfere with the efficacy of isoniazid?
Not to my understanding.
I had it in my head that it might, but
and just because of this is relevant to this group, given that we potentially prescribe, is that in the MJA last year they published some case studies of people getting optic neuropathy from vitamin B6 supplements, and I've had a few patients get peripheral neuropathy, not TB patients. They're just taking heaps of multivitamins. I've asked them to bring in their vitamins, and they're taking 200 milligrams a day. They bring in their shopping trolley of all the vitamins, and so it's quite interesting actually.
Yep, and people would be aware, as you would be, that the TGA, I think in October last year, put out a warning about pyridoxine in supplements, because if it's less than a certain concentration, then that doesn't have to be included in the ingredients of say the multivite, or in particular magnesium supplements. So people take magnesium for all sorts of reasons, and we're not aware that they're also dosing up on pyridoxine. So if you have a number of those supplements, yes, the risk is peripheral neuropathy due to pyridoxine toxicity.
I also wanted to ask, this is coming a little bit back to the acetylated status, is about whether there have been any trials, EBAs or early bactericidal activity trials or otherwise, that have looked at increasing the dose of isoniazid in fast acetylators to determine whether we can improve the bactericidal activity, but in drug-susceptible TB. I know we're talking about it in the setting of drug-resistant TB, but certainly if you've got someone who is metabolizing the drug quickly, so you have less exposure to the drug, could we not look at whether we could achieve higher drug levels? And therefore, I'm always thinking about what we can do to shorten TB therapy for drug-susceptible TB. I know there's a lot of discussion about MDR TB, but certainly more and more about drug-susceptible TB as well.
It's certainly been modelled.
Mm.
So some of the hollow fibre work from Tawanda Gumbo in Texas and others have modelled the impact of cetylator status on reaching, you know, PK/PD efficacy targets. So you're less likely to meet those targets if you've got a rapid acetylator status.
Mm.
But I don't know about any studies in patients.
Okay.
It might just be
So
interesting, Gail, just people may not be familiar with these types of studies, and that for TB drug development now, it's quite common for people to do this 1 or 2 weeks of monotherapy with a new drug, say bedaquiline and britomidine, and looking at the time to positivity and liquid media, and then they start their full treatment after the 2-week mark.
Yeah.
And
Yes.
so it's an interesting concept. We always worry that you're going to get drug resistance with monotherapy, but it's felt that for 1 to 2 weeks, it's actually okay. And then you can start the full course.
Yeah, certainly. But the group I worked with in Singapore, we did, I mean, not so much a question about the development of drug resistance, but, you know, when we looked at host-directed adjunctive therapies in tuberculosis, we did, I think it was at least 3 or 4 early bactericidal activity trials where we give patients with TB, the first 2 weeks, they receive, you know, certainly monotherapy, maybe higher doses of rifampicin, for example, or they receive celecoxib with allopurinol or other drugs, and we look to see what their TTP was within those first 2 weeks, so the time to positivity, as a marker to see whether there was any individual activity of that drug or synergistic activity if combined with, you know, monotherapy, let's say rifampicin, for example. So it's a tried and true method that's used in TB clinical trials. And, you know, some of the work that we did in Singapore was also whole blood bactericidal assays. That's a model that was developed by Bob Wallace he's now at the Aurum Institute in South Africa. But essentially, this is where you give healthy volunteers a drug, and then you know, at the various peaks, when the drug has, you know, kind of reached a certain level in their blood, you draw their blood and then you inoculate that blood with TB. So that's a whole blood bactericidal assay. And then you measure the amount of the effect that drug has on the inoculated TB. So, you know, there are lots of interesting models, I think, that are looking at how we can measure individual drug activity. Because often we can't get away with not giving TB patients treatment for too long. I think 2 weeks would probably be the maximum, but we do need to try to, you know, besides modeling, work out how these individual drugs work.
Can I just pick up on one of the points that comes out of that is you know, is it 1 week, as in this study on the katG mutated strains, or is it 2 weeks? If they'd continued this study for 2 weeks instead of 1 week, would they have seen any additional effects? They probably would have seen additional toxicity. And they postulated, look, the early bactericidal effect is well known of isoniazid, and they didn't really need to go longer than 7 days before getting onto a proper regimen. And look, it does highlight, you know, a lot of our decisions about, say, taking people out of isolation is based on the assumption that you have an isoniazid susceptible strain and that you're going to get a nice early kill, rendering someone non-infectious fairly quickly. And look, you know, isoniazid is a great drug for that, and it'd be remiss of me not to mention Dr. Tom Constantinos, who was previously the director of TB in Queensland, who my quote from him is, isoniazid, it's the penicillin of TB. So it's, when it's susceptible, it works really well.
Can I ask you a lab question? So, you know, traditionally we've only had the, you know, whether it's high or low resistance, if you like, based on presumably MIC. Now we've got the katG and inhA genotype, but could you infer with accuracy whether it's going to be high level, i.e. cat mutation, or inhA low level based just on the MIC that you might get?
You do get probably better data to look at that comes from the cryptic consortium. So you still do get some overlap. Most of the time it'll be correct. And look, sometimes you you may not have any of those mutations, but there might be some other mutations less commonly known that put your MIC up. So it's katG wild type, inhA wild type, but the MIC is high. But most of the time, if it's wild type to those, yes, it will be in the susceptible range. And similarly, inhA without any companion katG mutation, which sometimes occurs, or a sort of sequential mutation in the coding region. Yep, pretty much most of the time that's low level. So that it does correlate better than they stated in this study, but as I said, I've got concerns about their MIC measurement when they're saying that the lower end of the reference range was at the ECOF for drug susceptible strains. Was that the question?
Yeah, it was. Thank you.
How's the chat going? Because I haven't been keeping an eye on that.
I think that we haven't had much activity on the chat. So
see, Andrew Burke's corrected me that NomMen is a common PK software program, and I should have known about it. Thanks.
we won't keep people too long. So we're at 10 minutes before we're due to finish. So thank you once more, Chris. I think it was an excellent way to start this journal club. And thank you very much to everyone who attended. I'm very pleased to see such a robust turnout today. We will be applying for CPD CME points for this journal club, which I think will be very useful. April, if you don't mind sharing the link to the survey, please. It would be great if people could please complete this brief survey just to give us some feedback on what you'd like different about the journal club if you'd like to attend again. If you would like to attend again, I think it would be useful to register if you haven't already for the journal club. That way you can keep updated with when we're having it again. Our next journal club, just to pencil into your diaries, is planned for the 26th of July. The journal and the presenter will be announced closer to that time. So we're planning on having it every 2 months. We also have a few other activities planned on the ACNet Burnett calendar. This includes a clinical trial seminar that is tentatively planned for October. It will be all online with 3 to 4 speakers, all of whom have extensive experience in clinical trials in Asia Pacific. And Andrew, do you want to comment on the couple of other ACNet activities?
Yes, thank you. So we're going to have another sort of online webinar in about a month, actually. I haven't got the date, but Dr. Vicky Gray from Brisbane, Dr. Alice Sorka from Adelaide, also be running some 2 interesting TB clinical cases for discussion. And so I guess we have a forum like this. It's often hard to do discussions in this sort of forum on Zoom, but are looking for new ways of trying to, develop this model, if you like, for online discussions. And so, yeah, that clinical cases discussion will be 4th of June at midday, Australian Eastern Standard Time. Thanks, Clive.
Yeah, just to emphasise that it's going to be at midday normally, isn't it? It's just that I had clinic today, so I couldn't get here by 12.
We will, I think we can be flexible about the timing,
Right.
Because certainly we want this to be a regional initiative. Our plan is very much to have people from within the region present. And so that's the other thing I wanted to say. If anyone here from, Indonesia, Papua New Guinea, etc., would like to put their hand up to presenting, we'd love to have you present. The reason that we're having this journal club is because certainly it's modeled on the East London Journal Club, where they have, wonderful, robust discussions about journals. And it's just impossible for us to be able to attend because of the part of the world that we're in and the timing of it. So I certainly think that we can have equally important discussions about the literature. But because it's going to be a regional really effort, I think that, we can be very flexible about the timing. Yeah. Okay, well, thank you everyone once more. We'll end it there, 5 minutes to go. So I hope to see you at the next one. Just as a reminder, it's on the 26th of July, and I'll send it out through your emails. I will also plan to send a summary of the journal and Chris's presentation around for your own interest. Okay, thanks again.
Okay, thank you very much for having me.